Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific webinar titled Sex Differences in Fear Memory. I'm Liam Sanyo from the events team here at scientist.com, and I'll be the host for today's event. This webinar has been sponsored by Harvard Bioscience, so a big thanks to them for helping to make this webinar possible. We're very fortunate to be joined by Dr. Raül Andero Galí, who is an ICREA research professor at the Institute of Neuroscience at the Autonomous University of Barcelona. Dr. Andero will present research on the sex-dependent differences in the consolidation of fear memory in a rodent model. And with that, I'll hand things off to Dr. Andero. Raul, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you very much, Liam. And I also want to thank Inside Scientific and Harvard Bioscience for the invitation and all the staff organizing and promoting this event. Thank you very much to everyone, and also everyone from their homes or their offices joining this talk. Today in Barcelona, right now it's 4 p.m. We got some sun after some rainy days. And I will be happy to talk about sex differences in fear memory. So I'm going to go first slide. So my laboratory is in the Autonomous University of Barcelona. And besides the memory part that I will be explaining today, we also work on stress and memory paradigms, including animal models in rats and mice, and also humans, and in collaboration with nearby hospitals or hospitals in other countries. We also research on post-traumatic stress disorder in patients that suffer it, and one of our goals is to try to find better treatments for PTSD and potentially even being able to prevent it. So I have to disclose that I have a patent in which I may have conflict of interest in this talk. In my laboratory, we're specifically focused on fear, which is quite different from anxiety. And we can see in these cartoons that reflect perfectly well the idea, in which fear is a reaction of something that is going to hurt me right now, really bad. However, anxiety is it's about being afraid of something that may come or may not come, right? And I love the definition by Michael Davis, in which I was lucky enough to be in the same department as him in his last years before retiring at Emory University. And he was a great mentor. He still is. And I like how he defines fear and anxiety. And fear is a generally adaptive state of apprehension that begins rapidly and dissipates quickly once the threat is removed. However, anxiety is elicited by less specific and less predictable threats. And as you can imagine, the neurocircuitry of fear and anxiety are different, but they also have some overlap. And why my laboratory is interested in fear? Because fear-related disorders are the core of certain psychiatric disorders described in DSM-5, such as obsessive compulsive disorder, trauma and stress-related disorders, such as PTSD, as I mentioned in my introduction, and also as a general component of anxiety disorder, such as specific phobias or generalized anxiety disorder. So we understand that by understanding the molecular mechanisms of fear, normal fear regulation, and pathological fear regulation, we could treat many brain disorders included in this slide. Fear-related disorders are more prevalent in women than men. Women suffer around 2 times more often fear-related disorder than men. For example, PTSD, panic disorder, or obsessive compulsive disorder. So the reasons of this are largely unknown. There have been many authors pointing at biological or genetic or epigenetic situations that may lead women to be more vulnerable to suffering fear-related disorders. And I also think it's environmental. Probably women are exposed to harder situations across their lifetime that because we know like stress and fear are related, so greater exposure to stress may also lead to higher affectation of fear-related disorders. And another reason why my laboratory is focused on fear is because when studying memory, fear is especially helpful because the mechanisms are highly conserved across the species, for example, humans or rodents, and also because the work using Pavlovian fear conditioning for decades, so help us to understand what happens in the laboratory animals sometimes can be translated to humans. And within the fear circuitry, there are many brain areas involved in fear modulation. For example, the prefrontal cortex, the hippocampal formation, the amygdala, the bed nucleus of the stria terminalis, the hypothalamus, among others. And today, I'm going to be focused on the amygdala, which classically has been thought to be the center of fear. And nowadays, this idea is abandoned. And right now, um, we have an idea in which the amygdala is an important nucleus for fear, but it's not the center. It's just one brain area important. And I have to highlight that this amygdala-centric view of fear is mainly related to animal studies, because in human studies of fear, there are several papers and reviews and meta-analysis pointing it that sometimes there is no the amygdala is not so related to fear memory formation in humans, and other structures are. So I'm focusing today in the amygdala, but we have to keep in mind that is not the center of fear. It's one area that is highly involved in fear processing. And I'll explain you why we're so interested in the amygdala in my laboratory. As we can see here in this review, the amygdala in rodents has very interesting neuropeptide populations, and they are some of them expressed in certain areas, sub areas of the amygdala, like for example, in the central amygdala, the centromedial, centrolateral, capsular part, or BLA, medial amygdala, and there are many neuropeptides. And the reason we're focusing on TAC2 on the centromedial amygdala, it's because we, using this paradigm I'm going to show you now, we found in 2014 that the TAC2 neurons were necessary and sufficient for the formation of fear memories in male C57 mice. So the paradigm we use involves habituation to a context, experimental box, in which the animal, what basically does is to explore the environment. And then the day after, on the what we call the fear acquisition, an acoustic tone is co-terminated with a mild food shock. And this is done for 5 times. There are 5 pairings of tone and shock. So the more the tone and shock are paired, the more the animal freezes. Freezing, which is the absence of movement except those movements associated to respiration, denotes fear by the mouse, right? We cannot ask the mouse, "Are you afraid?" Right? So we measure fear by measuring freezing. So 24 hours later, we put the animal in the same box, but we change the environment, not only the color of the box, also the odor, the transportation to the experimental room. So we try to minimize the contextual cues that could make the animal remember that being in that room, in that box, is going to be dangerous. So we try to change as much as possible these contextual cues for the animal to be only afraid of the tone. And indeed, we're successful with that, because when we put the animal in the box, modified box, shown here in pink, the animal presents barely no freezing. However, when the tone is presented again and the shock is not presented, so the animal does freezing. And the more tone presentations, the faster the animal learns that the tone is not dangerous anymore, and then the animal does less and less freezing, as we can see here in the graphic below. So this is the paradigm I'll be explaining today during my talk. And in this 2014 paper, we found that giving a drug 30 minutes after fear acquisition, this tone and shock pairing, fear memory could be modulated. Many people has tried that before to modulate memory. And then how we evaluated that we were eliciting modulation of fear memory was to evaluate in the freezing levels during fear expression. And in the graphics below, you can see that given the drug not only 30 minutes before, but sorry, not 30 minutes later, but also given the drug 30 minutes before, 1 hour after fear conditioning, no, sorry, 10 minutes after fear conditioning, 1 hour after fear conditioning, but not at 4 hours after fear conditioning, we were decreasing the amount of freezing that the animal was doing on the fear expression test. So we're modulating fear memory consolidation by this drug, which is osanetan. This drug was given systemically. So what is osanetan? Osanetan is a neurokinin 3 receptor antagonist that is safe and well tolerated in humans. It was tested in the 90s for the potential treatment in schizophrenia in clinical trials, and also other papers have tried to have given successfully other neurokinin 3 receptor antagonists. So they have been given to hundreds of people. They're all of them, as far as I know, safe and well tolerated, and antagonize this neurokinin 3 receptor. And the receptor is what we call the TAC2 pathway, in which the TAC2 gene synthesizes the neurokinin B peptide that binds to the neurokinin 3 receptor, in which osanetan acts as an antagonist. And this NK3 receptor is a G protein couple receptor. In the mouse brain, we can see how the TAC2 gene is expressed in very few areas of the central nervous system, and one of the highest expressions is in the central amygdala, and most importantly in the centromedial amygdala, also as shown in the graphic before of the review of Dabrowska. Also, there is a lot of neurokinin B in the central amygdala, and there is high expression of the neurokinin 3 receptor, again in the mouse, and rat in the central amygdala and VLA. So what happens when we manipulate this TAC2 pathway? So during the years, we have tried different techniques, for example, with lentiviral overexpression by increasing the TAC2 expression of the gene. So what we found is that there was an increased fear memory consolidation by inhibiting the TAC2 expressing cells, all these in the central amygdala. So concordantly, we found decreased memory consolidation, and by stimulating the TAC2 channel rhodopsin expressing cells with optogenetics, we found increased fear memory consolidation. But all these experiments were performed in males. What about females? So when I started my lab, The first question we tested was, what happens if we give osanetan to not only males, but also females? So on the graphic in the lower part, in blue, we can see how giving osanetan in males replicated the effect of the 2014 paper I showed you earlier. And in females, we found the opposite effect. Given the drug systemically, enhance fear memory consolidation, as shown by higher levels of freezing during the fear expression test. So this is young adult C57 mice, and in females, we were not monitoring the estrous cycle in this graphic. This paper was published in Nature Communications, as Liam said earlier, and then although we didn't monitor the estrous cycle in the first figure, we were aware by other studies in which chronic NK3 antagonism of another drug different from osanetan but has similar properties, ESN 364, decrease testosterone and estradiol levels in males and in females, respectively. So we wonder, well, maybe the estrous cycle in females has an interaction with the effects of the drug, right? So indirect way to measure the estrous cycle, somewhat equivalent to the menstrual cycle, but obviously not the same to women. An indirect way to measure sex hormones is monitoring the estrous cycle with vaginal cytology, in which we get the cells, and then by looking in the microscope the cells, we can know in which cycle the animal is, and rodents have the estrus, proestrus, estrus, and metastrus. A direct way to measuring sex hormones, which we did for both males and females, is to make a small incision of the tail, not amputating the tail, not totally cutting the tail. We get a small incision on the tail, we collect blood, and this blood helps us to measure hormonal levels. I have to highlight that after habituating the animals to this procedure, this is not very stressful for the animals. Obviously, it's not. It's a little stressful, but it doesn't interfere with the results, with experimental results. So, when using the indirect method, doing the vaginal cytology to monitor the estrous cycle in females, and then we repeated the experiment I showed you earlier, and we gave the drug systemically, but we gave the drug only when the animals were in proestrus, estrus, metastrus, or diestrus, and we evaluated the day after the effect on fear expression. So we actually found out that the drug was only effective increasing fear memory when it was given in proestrus. So as a reminder, proestrus presents high levels of estradiol and progesterone. And obviously, because it is too much work to perform all the experiments in the 4 cycles, we refer to the literature, basically from Mohamed Milab's work and others, in which we use proestrus and metastrus as periods where the sex hormones are high, like proestrus, and metastrus are where the sex hormones are low. Obviously, there can be low levels of sex hormones in other cycles, or high levels of estradiol in other cycles, but somewhat we felt that in the literature there was like agreement that proestrus indicates high levels of sex hormones and metastrus lower levels of sex hormones. So we measure estradiol in females after fear acquisition, and mice 30 minutes after receiving fear acquisition, they receive osanetan, and we measure at 30 minutes. Sorry, there's a typo. It shouldn't be 330 here. 30 minutes after receiving the drug, we evaluated the estradiol levels, and osanetan increased estradiol levels only when animals were in proestrus during fear acquisition, but not during metastrus. That seems to be in line with the behavioral effect we got in increasing memory in proestrus by osanetan, increasing memory in proestrus, but not in metastrus. When measuring testosterone, we chose 2 time points because the dynamics of testosterone by literature appear to be different than estradiol and slower. And we measured testosterone at 30 minutes and 330 minutes after giving osanetan. And we found that osanetan reduced testosterone levels at 330 minutes, no effect at 30 minutes. And also in the amygdala, we were able to, by micropunch, we were able to collect tissue from the amygdala, and we also observed that concordantly to the results in blood, the testosterone levels were decreased in males. Technically, we tried obviously to measure estradiol with amygdala punches in female mice, and then we couldn't do it. As far as I know, there are no papers describing it. If anybody knows a method to do this, I would love to collaborate. We also measure progesterone. As I suggested earlier, progesterone can be high or low differentially depending on the estrous cycle. And we found no changes of osanetan on progesterone. And also we measure corticosterone, one of the stress hormones, to evaluate if osanetan is decreasing or increasing stress. And we found no changes of osanetan in these 2 hormones, discarding somewhat these hormones from a role of osanetan modulating fear memory. So in the next experiment, we said, okay, well, what we can do is to ovarectomize mice, right, and then remove all the sex hormones, right, that produced by the ovaries or the gonadectomies from males. But we are aware by literature that there's a lot of changes that are produced in the brain after ovariectomies and gonadectomies, right? So we thought, what about testing the compound osanetan in young adolescent mice with low levels of sex hormones just before puberty. So they have very low levels of testosterone and estradiol. And it's what we did. We gave osanetan on in the P28, and they were tested on fear expression and P29, and we found that osanetan did not modulate memory consolidation. So these results suggest that testosterone and estradiol are necessary for the sex opposite effect in NK3 modulation, and that progesterone and corticosterone are not necessary for these effects of the drug. So well, this was very interesting. But then in the next experiment, we wonder what happens with the cells expressing in the central amygdala after these hormonal studies. So the first question was, well, maybe we're getting this opposite effect because of the expression of the neurokinin 3 receptor. Is it higher or lower in males or in females? And so we evaluated the number of neurokinin 3 receptor neurons by immunohistochemistry, and we found that overall was similar across groups, including males, proestrus, metastrus, P28, and males and females. So we don't think the amount of receptor is responsible for this effect of azanetan in fear memory consolidation. Also, we evaluated the colocalization of NK3 with estrogen receptor beta and estrogen receptor alpha. We didn't evaluate the neurokinin 3 receptor colocalization with androgen receptor because we found in the literature papers suggesting that they were not highly colocalized in the central amygdala, and although there is some degree of colocalization, and we decided to tackle this NK3 receptor, androgen receptor idea in one of the latest experiments I'll show you today. So when we evaluated this colocalization of NK3 receptor with estrogen receptor beta and alpha, we found that there was no changes between males and females, but indeed there was a decrease of estrogen receptor beta in proestrus and metastrus compared with males. And we use this experiment again for giving a drug, a dual estrogen receptor agonist, at the same time as a azanetan, and that will be the last experiment I'll show you to follow up on this immunohistochemistry data. All right, so now we look at what happens at the central amygdala cells, and then we also gave the drug systemically. But what happens when we give the drug intracentral amygdala, or when we modulate the TAC2 expressing cells with DREADS, with chemogenetics? So in the 2014 paper, we found that giving azanetan in the intracentral amygdala, what it did was to decrease the amount of freezing during fear expression test, suggesting impaired memory consolidation, which is consistent with the effects on the systemic drug. So we did the same, but now in females in the recent study, and we found the opposite effect, which is concordant with the systemic injection. We found an increased memory effect when evaluating the freezing levels during fear expression in females, specifically in proestrus. Also, we use TAC2 Cree mice and Cree-dependent DREADS to temporarily silence the TAC2 expressing cells in the central amygdala 30 minutes after fear acquisition. We use the same timing as giving the drug. So the way we temporarily silence these neurons is by giving CNO, which reduces the activity of these TAC2 cells, silences cells for between 6 and 8 hours after the CNO is given. So what we did here was to test if we could replicate the findings given the systemic injection of azanetan, and the answer was yes. By temporarily silencing the TAC2 expressing cells in the central amygdala in males, fear memory consolidation was decreased, and however, it was increased in animals in proestrus during in which the CNO was given in the fear acquisition test. However, in the animals in metastrus, when we silence these TAC2 cells, there was no effect on memory consolidation, and that was very, very interesting. Of course, the next thing we did was to measure the amount of TAC2 cells there are in males and females, and we found no differences between them. And I think this is concordant to the immunohistochemistry studies of the neurokinin 3 receptor, showing that the levels of NK3 and TAC2 are similar between males and females, and that they may not be the reason why we have a sex opposite effect on memory when giving azanetan. Further, what we did was a micropunch after fear acquisition, and we evaluated by qPCR 32 genes, and we performed bioinformatic analysis that showed we evaluated mRNA levels after fear acquisition, and what we found was that the AKT pathway was upregulated in proestrus, and other members of this pathway were downregulated in males. So we further explore this hypothesis of mRNA now with protein, and we look at several proteins at 10 minutes. The mRNA data was at 30 minutes and the protein data at 10, and we look at several actors of the AKT pathway, but also we look at CREB, GSK3 beta, mTOR, and beta-catenin, which have been involved also in memory formation with several paradigms and different brain areas. So interestingly, we found in males a downregulation when osanetan is given on the total protein divided by GAPDH of GSK3 beta. And in females, we found an upregulation of the total protein of AKT. And we found a downregulation of beta-catenin when osanetan was given in females in proestrus. However, for example, CREB that has been widely, and mTOR that have been widely implicated in memory processes were not affected by osanetan. We are not sure that CREB and mTOR are not actors in this effect of osanetan in memory, but maybe it's the timing we chose. We didn't try other timings, but it would be interesting to replicate these experiments at different time points, and maybe CREB and mTOR maybe also presenting changes between their groups. So the pathway that we believe is modulating the osanetan and then neurokinin 3 receptor involves the phosphorylation of AKT, which inhibits the phosphorylation of GSK3 beta, inhibiting beta-catenin, leading to memory consolidation. And we're basing also our results in another paper of fear conditioning published in 2011, basically focused on beta-catenin. So in this final experiment, as I've been telling you, what we did was to give osanetan systemically 30 minutes after fear acquisition, and also intracentral amygdala, we gave drugs to test whether this receptor or these pathways we're interested are responsible for the osanetan modulation of fear memory consolidation. Specifically in males, we gave an AKT activator. In color are the drugs that they were given intracentral amygdala. And as you can see here, all groups receive osanetan, but intracentral amygdala, they did receive vehicle, AKT activator, or androgen receptor agonist. So when male mice receive the AKT activator or the androgen receptor agonist, they present higher levels of freezing during the fear expression test, suggesting that this pathway, the AKT pathway or the androgen receptor agonist, are necessary for the modulation of memory by osanetan. In females in proestrus, we gave an anti-AKT 1 and 2 and a dual estrogen receptors antagonist. So it's an antagonist for both the alpha and beta. And memory was decreased when we deliver intracentral amygdala, the anti-AKT 1 and 2, and the estrogen receptors antagonists, suggesting that they're important for the regulation of osanetan in memory. So as a summary, with our data, we can suggest that the TAC2 pathway antagonism in the central amygdala involves the neurokinin 3 receptor, and specifically in males, it decreases testosterone levels, the PI3K, AKT, GSK3 beta signaling, and memory. However, in females, Sanitan increases estradiol, molecular pathways proteins involved in memory signaling, leading to enhanced memory. So with this, I would like to thank my lab, and especially Antonio Florido, that is the first author of the publication. So if you have any questions, I will be happy to answer them. Thank you very much for your attention.
Fantastic. Well, thank you, Dr. Andero, for a great presentation. And with that, we'll move right on to the Q&A. All right, first question here, Raul, is a great one. What are the implications for menopause? Could certain drugs affect menopausal women differently?
Yeah, it's indeed a great question. I didn't have time to go through this, but neurokinin 3 receptor antagonists are being tested right now in clinical trials by several pharmaceutical companies for the treatment of hot flashes in menopause, mainly because its effects on estradiol, right? So it seems like it's going to be approved relatively soon. And also, what we suggest is, besides this beneficial effect of treating hot flashes, also they could help to improve memory, because it has been shown in some women with menopause that there may be problems in memory. So the approval of NK3 may also help not only in hot flashes, but also increasing memory.
Great answer. Um, so should we also be worried now about other drugs that were already approved and are available at the pharmacy now before, for example, this testing might not have been done on them if they're maybe the drugs were only tested in males, for example. Could drugs that are currently approved be affecting humans in a similar way?
Mm-hmm. Well, the first thing is, like, we don't need to panic. Obviously, all the drugs we have in the pharmacy are very good. They go through a lot of controls. But what is true is, like, for clinical trials, obviously they're tested right now in men and women, and the effects are controlled. However, a deeper understanding of a specific activity of drugs in men and women could be beneficial for everyone. Instead of trying to find a drug that works for everyone, maybe we're leaving behind some drugs that are amazing for men or women, right? That's one point. And the other point is that, although this has changed or is changing, I want to say, is that preclinical models using rodents generally used to use only males. Right now, most of us are using both males and females, but a lot of the knowledge generated for the last decades was only tested in males. So maybe with these preclinical experiments performed many decades ago that led maybe to the test of drugs in people, we discarded, or we didn't test drugs that could have been helpful also in women, because females were not tested, right? So we need to be aware of this and to improve the system to find better treatments for everyone.
Yeah, that's a really great point. I suppose it's a good thing that a lot more regulatory agencies are requiring researchers to use both male and female mice. So
Yep.
hopefully that kind of thing doesn't really happen too much anymore. All
Mm-hmm.
right, another question here that I think refers to slide 25. Is the 24-hour time point critical? And Raul, do you think your results would change with a longer term time point, like 48 hours or 72 hours?
We haven't tested the animals later. We believe the answer is no, because fear memories last for very long in a mouse. So, for example, if you do fear conditioning, the fear acquisition paradigm, and you test the animals 1 month later, they will still remember the tone and they will still freeze, right? So it's a very strong memory. What is true is that I mean, we couldn't control for the estrous cycle at the moment of fear expression, right? But it kind of controls itself, because if the animals are in proestrus on the day on the fear acquisition, so most of the animals are going to be in the same on the same estrous cycle during the fear expression test. So the answer is, like, I think results would be the same if we test the animals on 1 week than 1 day, but it would be a very interesting experiment to know, and we won't know until we test the animals for sure. Yeah.
Definitely. Um, all right, next question here. Why did you choose to inject the antagonist after the stimulus was given? Isn't it too late to administer an antagonist, 30 minutes after the onset of the neural changes? And do you have any data on the estrus cycle synchronization of mice from the same cage?
Mm, okay. So yeah, the reason to give the drug 30 minutes later, it's because, as I said at the beginning, one of our interests is would be to be able to treat and prevent PTSD, right? So although we're using healthy animals, if you allow me, and our results, if they can be extrapolated with you, you never know, right? Because an animal is very different, of course, from a human. So ideally, we would like to have a drug that we can give in the emergency room of somebody that had, for example, a car accident, and that drug may decrease the consolidation of that traumatic memory, decreasing the probability of that person to suffer psychiatric disorders, right? So that would be the idea. So, and also because we tested the compound, we gave it 30 minutes before fear conditioning, and we found the same effect as giving it 30 minutes after or up to 1 hour. We thought that the effect was going to be more, if you allow me to say, clean, because we give the drug during the consolidation after the animal has been exposed to the box, and we believe this is the best way we had to modulate memory consolidation after fear acquisition, right? And in regards to the second question of the estrous cycle synchronization, so we did try to synchronize the estrous cycle of the animals on the same box. We couldn't get to do it. And actually, this is not part of the paper, because we didn't, we didn't look at this and make notes and such. But like I consulted other colleagues, and they told me that they also couldn't synchronize, and actually would be ideal for us to synchronize the estrous cycle of our minds. But they told me that they couldn't do it either. Like we also tried to put in experiments that were not part of this paper, of course. We did try to put male urine in the cage to help females to synchronize the cycle, and it didn't work. So actually, my opinion, my personal opinion, although it's a little probably I would need to read more. It's like maybe the classical experiments in which the estrous cycle synchronization was performed and described, it was in older vivariums, which are very different from the ones we have right now, in which we have ventilation 24/7, air conditioning, controlled temperature, and also they were probably back then less crowded, so the conditions are very different, right? So I consulted 3 or 4 labs, and all of them told me, we wish we could synchronize the estrous cycle, but we couldn't make it happen. So the same for us. We couldn't make it work. And actually, if somebody knows how to do it, we'll be happy, because we tried everything we found in the literature and it didn't happen. So yeah, very, very interesting question.
Yeah, definitely an interesting research avenue to maybe explore. All right. Another question here in terms of about the translational side of things, but is the NK3 receptor expression in the central amygdala similar between mice and humans?
Mm-hmm. Yes, the expression of NK3 in rodents and humans is somewhat similar. It's expressed in also in emotional areas of the brain. Is I would say it's not exactly the same, but it's somewhat similar, right? So yeah, it's expressed in the amygdala, the BNST, in the cortex. So yes, also the pharmacodynamics between rat and human of the of Sanitan are somewhat similar. There's no data on mouse versus human, but we think that yes, there's lots of similarities on the TAC2 pathway in mice and rodents and humans. Although I'm aware there are reports showing like for example, in rhesus macaque, there's a study showing the expression of NK3, which has some differences with mice. There's like some differences, some similitudes, but I think like the fact that NK3 is also expressed in humans in emotional areas such as the amygdala and the BNST, and that NK3 receptors antagonists are safe and well tolerated. I think it's there's a high likelihood that they could modulate also memory in people without side effects. The only side effects would be a temporal decrease of sex hormones that could be uh, taken to normal levels by taking another drug. And also, there could be therapeutic interventions in which modulating testosterone or estradiol could be also beneficial. So there's another line of interest for NK3 antagonists just by modulating sex hormones.
Right. Yeah, great answer. Um, Another interesting question here, more about the fear conditioning process. Um, So this SND says, hello, Raul, great talk, very interesting. I have a more general question. There's some debate whether males and females express fear in the same way. Do you think assessing the freezing response during fear conditioning is the best manner to compare the fear response in both sexes?
hmm Yeah, we're aware of that, and I think that's mainly for rats. My personal opinion, that's more for rats. That I'm aware that female rats do darting behavior. In the supplementary figure, we evaluated darting behavior in our mice, and they barely do darting. So my personal opinion is that rats, as being a more complex animal than a mouse, they do more behaviors, right, than a mouse. And then, for example, darting can be very helpful in rats. And in mice, I think maybe the other behaviors may not be that relevant, although, we would need more studies to assess it. As again, and when we measure darting in our experiments, our animals did not dart. And yeah, I think, and also freezing, it's been a reliable measure of freezing in males and also in females. So yeah, I think it was the best thing to do for our study. Yeah, good question too.
Yeah, great point. Um, So in humans, a common issue for some PTSD patients is the relapse of fear. Do you know if did you do any fear relapse or fear reinforcement experiments after giving the antagonist?
No, we didn't do it, but it would be very interesting. Yeah, and as pointed, yeah, it would be really, really, really good to try, right? To do the fear conditioning test, give the drug, test the animals and fear expression, and then wait for maybe 1, 2, or 3 weeks and test the animals again. Yes, it would be very interesting experiments to perform. Yeah.
Yep, definitely. Um, There's a question about uh, the you're talking about how the drugs didn't affect adolescent mice before their levels of sex hormone were very high. Could that have could the drug not have worked because their emotional circuitry is still in the developmental stage, or is it definitely related to sex hormones?
Mhm. What we know is that at that age, at P28 and P29, they already can acquire fears. In the graphics, you can see that they acquire fear. Of course, the fear circuitry can be different, right, from adults. Yes, that could be a possibility that we cannot discard. But yeah, also the amount of learning of the fear learning they got, and also that the expression of NK3 of in P28 and P29 was similar to adults, make us think that it was the best solution. I mean, in the end, there's no good way to assess this. If you want to try like to give the drug without sex hormones, the other alternative was gonadectomies to remove the gonads, which has wide implications of the brain. So yeah, no matter what you choose, you will always open more questions. But yeah, very very interesting.
Yeah, that's a great point. Another good question here. So, do you know if the effect is specific to fear memory? Have you examined any sex differences with response or in response to this drug during a different learning exercise, like novel object exploration or other tests of memory?
Mm-hmm. Yes, actually, we're working right now. We have a paper submitted looking at the effect on oxanethan on novel object recognition, and results replicate the data on fear conditioning. So we believe that the effect of oxanethan in memory is not specific to fear, but it can be expanded to other memories such as novel object recognition, in which the prefrontal cortex and the hippocampus have been shown to be very important. So we believe that it's not only this drug affecting the neurons in the amygdala, maybe, although I'm hypothesizing because we haven't manipulated that and we haven't gone that far on the novel object recognition studies, but it could be like this memory modulation. It's not only happening in the amygdala, but it can happen in other structures, but it always goes in the same direction, decreasing memory in males and enhancing memory in females in proestrus when there are high levels of uh, estradiol.
Fantastic. Um, Interesting question here. Does estrogen affect NMDA receptor expression in the amygdala?
Mm-hmm. Yeah, I'm not an expert on NMDA receptors. I believe it does affect estrogen and NMDA receptors, and then yeah, it could be one reason why our results are different between males and females. And I think in the paper, I'm not sure if we look at NMDA or if we did immunohistochemistry, but yeah, I'm sorry, I'm not an expert on NMDA.
Yeah, no problem. Another question here is sort of about the human side. Would you expect women using hormonal contraceptives to react similarly to the low estradiol group?
So hormonal contraceptives, yeah, so to the low, yeah, so to the low estradiol group, to, yeah, so it would be like the equivalent to metastrus, I understand. Yep. Yes, yeah, I would think so. I would think my hypothesis would be like maybe the drug wouldn't work on women, maybe taking hormonal contraception. And also one of the implications, probably the main implications of this study would be also to highlight that the same drug could given in completely different contexts to men and women. Because in men, we would give the drug to somebody that just had a car accident and is in the emergency room, whereas this drug should never be given to women, because then we would increase the memory of the traumatic event. So we would give the drug to a woman that has gone to psychotherapy, and after a successful therapy intervention, after one successful day doing therapy, then we want to increase that memory, right? But we would never do that to a man, because then we would impair that memory consolidation, right? And also, the last thing I want to highlight is that we don't want to erase memories. I've been asked this a lot. I don't want to erase memories. I want to modulate slightly decrease or increase. If we want to erase memories, you would go to completely different approaches that are not my goal, although interesting.
Fantastic. Yeah, great answers. I think in the interest of time, um, we'll have made that the last question. But Raul, thanks so much for sharing your insights with us today. It's been a real pleasure having you with us.
Thank you, everyone, and thank you, Liam.
Yep, definitely. Big thanks to the audience here for participating. And last but not least, we'd also like to thank the sponsor, Harvard Bioscience. So in closing, we hope you enjoyed this Inside Scientific webinar, and we'll see you again next time. Have a great day, everyone.